Hello friends, Ace Botany. Next episode. In this episode, we'll be discussing under quick reference NCERT, third part, sexual reproduction flowering plants, microsporangium, and wall layers. Meanwhile, short question, I am daily putting two, one from first year, one from second year. Detailed notes of NCERT Botany, they're already in YouTube. So subscribe Ace Botany, you'll be getting all this. For convenience, I will share the screen so it'll be easy for you. I think screen came, so we can proceed. So NCERT quick reference, Anther Wall Layers. All references based on NCERT March 2024 reprint edition. And first page you can find 4 sets of diagram all concerned with the anther in angiosperm and even cellular layer, that is this one. It's a usual labeling question. So we can continue with the second page, all NCERT. Okay. Now we'll be proceeding with the different layers of an anther wall layers, microsporangium, etc. So structure of microsporangium. So when we come to structure of microsporangium, it is first it starts with the shape. A typical microsporangium. Typical microsporangium appears to be circular in TS. Here you can find this one. Circular in TS. Outline is circular. And where it is located? Location is at corners. Corners of each anther lobe. Corners of each anther lobe. Each anther lobe. See, this is a corner. This is a corner. So each anther lobe means 2 lobes. Each lobe, 2 microsporangium. These are wall layers. So total 2 into 2, 4 microsporangium. So 2 in each lobe. Likewise, 2 lobes, anther lobes. So 2 into 2, 4. That is why a technical term, tetrasporangiate. Sporangiate. I think idea clear. Tetrasporangiate. How it become 4. Now each microsporangium is surrounded by 4 wall layers. 4 wall layers. What are the which are that is epidermis, endothecium, middle layers, tapetum. So these are layers. Inside the tapetal layer, we can find sporogenous tissue. I think idea clear, diagram clear. Now outer 3 wall layers connected with the protection helps in dehiscence of anther to release pollen. NCERT page number 5. I think first page is clear. Now we can proceed to each wall layer. First is epidermis. Diagram very clear. NCERT enlarged. Now it is the outermost layer. Outer layer. That is epidermis. Normally, it is single layer. Normally you can find single layer. Main function is protection. Main function is protection. Now, after epidermis, inside we can find endothecium. Endothecium is just below epidermis. Just below epidermis. And it helps in, function is, helps in dehiscence of anther. Opening, dehiscence of anther. So this is epidermis, just below you can find endothecium. And how it is helping? Principle is hygroscopic action. What absorption releasing? Hygroscopic action. Similarly, these cells have alpha cellulosic band. Cellulosic bands. Fibrous in nature. Fibrous in nature. And they arise from, all important, they arise from inner tangential walls. That is regarding endothecium. Now comes next layer that is given here, middle layers. Middle layers ranges from usually 1 to 3. Usually 1 to 3. And it is ephemeral. What is meant by ephemeral? Ephemeral means short-living, not long-lasting. And what happens to it? It will degenerate at maturity. That is why it is ephemeral, maturity, and function is supposed to be protection. Now comes innermost wall layer. That is tapetum. Here we can find tapetal cells, innermost wall layer, tapetum. That is innermost layer. That's important. Innermost wall layer, tapetum. And tapetum, inside the tapetum we can find, or inside the tapetal layer, tapetum will surround the sporogenous tissue. See this one. These are the sporogenous tissue. So this is tapetal layer. I think I did clear. And the function is nourishment. To which cell? Nourishment of developing pollen grain. Developing pollen grains. Pollen grain, write in full, otherwise confusing for you. Nourishment for developing pollen grain. And these cells have some abnormality. In which respect? One is nucleus. Number of nucleus will be more. And second is amount of DNA, that also will be more. That is tapetum, nutritive layer, having some abnormal property. Now we can proceed to next sporogenous tissue. Location, center of each microsporangium. Center of each microsporangium. That is where it is located. And is clear in anther tears. Young stage also it will be clear. Now feature, it is compactly arranged. Homogeneous cells. And sporogenous tissue, that is this one. These are the sporogenous tissue. Sporogenous tissue. Cells undergoes meiosis to form microspores. Later, pollen grains etc. will be produced, that we will be studying in next episode. So mainly dealt with the different wall layers and microsporangium location, etc. If you like this episode, subscribe or like it, share it, comment on it. We can meet in the next episode with the further aspects. So take care. Bye. Thank you.